Konstantinova, who is co-founder and chief scientific officer at VectorY, will be presenting reduction of oxidized phospholipids or misfolded protein aggregates by AAV-VecTabs in an ALS preclinical model.
Thank you very much. Can you hear me well? And do you see my screen?
Not the screen yet, but I can hear you.
Do you see it now?
It's coming. There it is.
All right. Okay, thanks a lot indeed for the introduction, and I was the one asking the last question because
just mention one thing. It's not in slide mode. I don't know how.
I will do it in a second. I just wanted to be sure that you can see the screen. So now you see it in a slide mode, I hope.
Yes.
Yeah, so thanks for the invitation and for allowing us to talk about our new programs in ALS. So we are also a gene therapy company, and I just realized we are doing something very similar as what Sola presented. So I will have a bit of an easier task to explain what is gene therapy and also what do we want to target. So we are gene therapy company based in the Netherlands, in Amsterdam. We are only 2 years old, so we're quite young, a biotech, and we have a management team that's been founded mainly based on broad experience in the gene therapy fields, both in the scientific space, but also in the development space. So our two main programs are in also in ALS, in Huntington's disease. So the first program that we are developing for ALS is not based on DPhil, it's based on oxidized phospholipids, which I will explain in a second. And the second program is also targeting DPhil aggregation. Besides using gene therapy with AAV, I didn't mention we are also focusing on expressing vectorized antibodies, similar to the colleague before me. And then we will have some specifics there. Our company is also making a big deal out of capsids. So we are developing capsids technology, trying to improve the AAV capsids, and also trying to improve our vectorized antibodies, which we call VecTabs, by adding specific tags that lead to the degradation of the antibodies. And last but not least, we are also aiming to have our own manufacturing facility on a short basis so that we are able to scalably manufacture the gene therapy products. I will not talk about the technologies today. I will only focus on the ALS programs. I will also not talk about Huntington's disease, although I myself come from the Huntington field. And I have to say, I've been working on Huntington for almost 12 years and seeing how rich ALS is and how many clinical trials are here relative to Huntington is really quite new experience. And I really hope we can make a difference with our ALS programs. So we are targeting sporadic ALS also with our two products, also for the reason that ALS, sporadic ALS have a different pathogenesis relative to the familial ones. So our first product, as I said, targets oxidized phospholipids, and the second one targets misfolded TDP-43, and there is quite some evidence already in the literature showing that the two phenomena are largely linked. So we don't mutually exclude those two processes from each other. However, we are developing those two products separately. So very recently, actually, Wong et al. Published really a month ago in Brain Communications, something that we've been thinking about in the last year and a half. And that's mainly that sporadic ALS is hallmarked by accumulation of ox-LDL and oxidized phospholipids that are in a complex with ApoB. And actually, if you inject this CSF from sporadic patients in mice, the mice, first of all, develop ALS pathology, but also very importantly, they develop TDP-43 pathology. And that's actually the slide from this paper. And Merit Cudkowicz is also a co-author of this paper that demonstrates a huge motor neuron loss when only 4 microliters of CSF from patients is injected in mice, in wild-type mice. And also very interestingly with that, this is associated with increase of phospho-TDP-43 if you look at the graphs on the right side. So that supported our hypothesis that oxidized phospholipids are a very upstream target for ALS, and that they also to some extent are linked to phospho-TDP-43 aggregation. So that's why our first program is indeed in targeting lipid toxicity with gene therapy, and then the second program targets phospho-TDP-43 in the soma of the motor neurons, but also in the neuromuscular junction, and I will show you some data that we have accumulated there. So for the oxidized phospholipids targeting, we are using actually quite old antibody, or originally. It exists already for 30 years. It's called E06. And interestingly, this antibody was used to stain ALS patient sections, motor neurons, and to demonstrate that motor neurons of patients are really covered with oxidized phospholipids. That's also work from Munoz from this year, so quite new evidence. Also what they showed is that those axons or those neurons are covered with oxidized phospholipids, and also they coincide with the damage marker APP. What we know from actually our scientific advices and our collaborators about this antibody that we are using and the targets is that oxidized phospholipids gets accumulated during many pathological processes in the periphery, but also in multiple sclerosis and most recently also in ALS. And the process is typically caused by shortage of the phosphatidylcholine tails that results in a flip out and exposure of new epitopes that are then targetable with this E06 antibody. Again, the E06 antibody only would bind to oxidized phospholipids, and it would not bind to their non-oxidative form. So we took this antibody, we have optimized it and humanized it and also vectorized it, which means that we also, similar to solo therapeutics, we cloned it as an expression vector in an AAV virus, and then we deliver this to mice, and eventually we want to deliver this to patients with an intracerebral administration. So as you can imagine, the essence of our work and the targets that we have precludes the use of the classical genetic models, at least for targeting oxidized phospholipids. Of course, we looked at the SOD mouse model, but first of all, it's a genetic model. It does not have a really increased OXPC there, and also it does not have TDP-43 pathology. And these are the main pathologies that we would like to target. Therefore, our company has focused largely from the very beginning on using iPS-derived patient cells from both sporadic and familial patients. And we also generate neuromuscular junctions on chip in-house. And we also collaborate with academic partners to test our gene therapy on on organoids, and we are performing transcriptomic and proteomic analysis based on for efficacy readouts. Of course, we know that there is no way to go around without animal studies. So the biodistribution studies we are doing in pigs, not in monkeys. And I will also show you a little bit about that. So the E06 antibody protects from motor neuron toxicity. We have been able to demonstrate that by adding oxidized phospholipids to patient-derived ALS motor neurons, and then looking for the effects, for the preventive restorative effects of the E06 VectorY antibody. So we have been able to demonstrate that, but furthermore, we have also been able to demonstrate on a transcriptome level that when we treat healthy motor neurons with oxidized phospholipids, we are generating an ALS-like phenotype with ApoE really highly increased, which is also one of the hallmarks of motor neuron pathology. And more importantly, when we treat those, let's say, damaged motor neurons with gene therapy with E06, we can restore this transcriptome profile. So that's what we have shown on transcriptome level. The next question that we asked is if oxidized phospholipids can also induce TDP-43 pathology, as has been shown in the literature. And what we are showing here is that the red is the non-oxidized phospholipids and the pink is the oxidized phospholipids. And this graph represents phospho-TDP-43 aggregation. So what we have been able also to show on a more phenotypic level is that by treating ALS motor neurons with oxidized phospholipids, we can also induce TDP-43 pathology. And the last question we asked is there an overlap in the transcriptome between the genetic forms of ALS and also the healthy cells that we treated with oxidized phospholipids? So POMPC is one of the most enriched oxidized phospholipids in the brain. And the answer is yes. So we looked at 1,600 ALS-related genes, and we have been able to demonstrate on RNA level initially that quite a lot of them are dysregulated in accordance with the ALS phenotype, and for some of them we also have been able to validate that on a protein level. As I said, we are doing gene therapy. So one of the major gene therapy is a single treatment, at least with the AAV. So one of the major questions is also, can we reach the right brain areas? Can we transduce enough brain cells to expect efficacy of the gene therapy? So we have injected pigs either intrathecally, that's on the upper part, or intracisternally on the lower part, and have looked at distribution of our AAV capsid throughout the CNS of the pig. So what we have selected is an injection route that will be an intracisternal administration, because there we see a very nice cortical distribution of the gene therapy, and also a good enough spinal cord and deep brain structure distribution. So this will be our injection route for potentially for both programs. So the second program that we have in-house is targeting mislocalized and misfolded TDP-43 protein. So that's a bit more familiar target, of course, and I'm really excited to hear so many good talks about UNC13A, statin, progranulin as potential targets as well for ALS. We are targeting TDP-43 aggregates, and also what we have been able to show in our work is that we are able to restore the lack of nuclear TDP-43 or the loss of nuclear TDP-43 function at the body of the cell, but also at the neuromuscular junction. So we have designed quite a few single-chain variable fragments, both intracellularly or intracellularly expressed. Our hypothesis at the moment is that for TDP-43, we would like to stay intracellularly and target the intracellular TDP-43, but we also have versions that have secretion signals that would be allowing for secretion of the antibodies outside of the cell. So again, the gene therapy concept on the right of VTX2, it's AAV5 vector that's injected to deliver to motor neurons. The antibodies are being continuously made, bind to the aggregated TDP-43, and then clearing it through the cellular system. This is the outcome of our initial screen. So we have screened 26 vectorized antibodies for aggregate reduction. And you can see that quite a few of them are really good in reducing aggregate reductions in the cellular system. And from those 26, now we have selected 11 that are the darker brown ones, darker gray ones that are going to be tested now in an AAV format for efficacy in patient-derived iPS cells. And eventually, at the beginning of next year, we are also planning to use exactly the same model, the doxycycline regulatable model, for testing our gene therapy. In our case, we will be injecting at 5 weeks of age, because we think that this is the age when it would be more representative for potentially disease progression. A little bit about our neuromuscular junction and the specificity of the VecTabs to TDP-43. What we have been able to show is that we are specifically clearing TDP-43 aggregates in patient-derived cells. And we are building those neuromuscular junctions in-house. And we also have shown that in the axons of the neuromuscular junctions of the motor neurons, where we have a co-localization of the VecTab together with the phospho-TDP-43, we are able also to reduce specifically phospho-TDP-43, which is the hallmark of ALS, and also gives us confidence that we are very much on target. And the last question we looked at is, it's known that there is a reduced TDP-43 in the nucleus in ALS motor neurons relative to the healthy ones. So what we have shown is that when we sequester the cytoplasmic aggregates, the TDP-43 aggregates, we're also able to restore the nuclear levels of TDP-43. So this is almost my last slide. Of course, we're thinking to go to the clinic, and I'm not a clinician, but we have already had a scientific advisory board that have advised us to have a placebo study, even with gene therapy, and start with a small number of patients, because the gene therapy is reversible, and have 6 patients treated in the low dose, and 12 patients treated in the high dose. And then the primary readouts will be, of course, efficacy and tolerability of the gene therapy. But of course, we are going to look at all the biomarkers that were discussed today and yesterday. And ALS-FRS will be used as an exploratory biomarker. As I said, many of us, this is always a group effort. It's been a huge amount of work from the whole team. So we are in Amsterdam in the Science Park, 65 and more, actually more than 65 people at the moment. And we're quite international. So there are about 15 nationalities at VectorY. So I want to thank you for your attention, and I'm open for questions.
Thank you. Both of your programs are fascinating. You covered a lot of territory. A couple of questions. I'll start with the lipid toxicity mechanism in EO6. I noticed it's an IgM, which is a little bit atypical, at least in my understanding, for therapeutic antibodies. Have you explored the idea of an IgG that would target, or is it that the phospholipids are such a broad target space that the IgM is actually well suited?
Yeah, thanks for the question. So the IgM antibody was originally isolated 30 years ago from patients with cardiac infarctions. And that's why originally IgM has been used, and it's actually used by our collaborators from UCSD. But we are using a single chain variable fragment, so we are not using the tail of the antibody at all. And we have tested in the lab at the beginning both IgMs and IgGs, and they both work as well. But for our gene therapy purposes, we will be using single chain variable fragments.
Thank you. And then a question on the TDP targeting. So you demonstrated in the cell-based assay the reduction in cytosolic TDP43, a reduction described as reduction in aggregates. Have you looked at cell survival in those? Because that's in a stress context. Does it rescue a cell death phenotype in any way, or have you looked at that?
Yeah, definitely. So apologies, I didn't show it. This is one of our primary readouts, cell survival. So for both programs, actually, we are showing dose-dependent protection, or we call it rescue of motor neuron toxicity with the candidates that we select to continue further development. So yeah, this is one of our primary readouts. Yeah, good point. Apologies.
Well, thank you. We're running a tiny bit behind, so no worries. It was a great presentation. Thank you.